which we have done also in the last years. We try to find in some specific fields 3 or 4 people who will tell us about the challenges to push the limits of nanomedicine and targeted medicine. In this year, we felt that on one hand, where's the bad boy? On one hand, we would like to listen to a realistic view, as Ehud Gazit calls it, regarding nanomaterials for molecular theranostics. The second one was that we said this reprogramming is so interesting, yet we learned also that it was done a long time ago already at the lab of Michael Zela. And the third one, I think, is very important. We found someone at IBM who I think is an excellent speaker because he is very open-minded. And I said, can you do never, never, never a promotion talk like we had it 3 years ago on IBM systems, on Blue Jeep or anything else? But could you give us a view how big data will influence, or the fact that big data is influencing the near future of medicine, how we have to see it and to show a view, maybe even into the future. So now I'm very glad that Udi, you who called yourself the bad boy,
bad boy.
would give us the first talk, and the floor is yours.
Thank you very much, Beat. It's really a pleasure to be here. I feel that our advancement in providing you paradigm for medicine and nanomedicine is following the CLINAM. So 2 years ago, I told you about the first clinical trials for a modulator of interactions at the nanoscale. Last year, I was speaking about new hybrid of DNA and peptide, and this year I would want to spend quite a lot of time on new paradigm for metabolic disorders related to interactions at the nanoscale. So indeed, our group is working on things that are ranging from materials to energy storage to flat panel display, some of the IT things, but also things related to medicine and nanomedicine. So how can we do all of this? Our interest is really in molecular self-assembly and molecular interaction. We are interested in the way that molecules organize to form ordered structures at the nanoscale. As related to biology, our attempt for some years already was to try to define the minimal recognition elements. That is, understanding, looking at molecular system and look for those structures that can mediate the formation of well-ordered structure in a reductionist approach. So we take the chemist approach to look into biology, and we got to smaller and smaller structure. I spoke about it previously in CLINAM, so I will just mention in briefing. Also on the extension to non-biological structures, I mentioned a few years ago. But we make all kinds of architecture in the nanoscale, including those that are related to diseases, especially degenerative disorders. And our range in biology range from the largest self-assembled structures are viruses, and the smallest are metabolites. And some just highlights from this is from Nature Communication. Just a few months ago, we demonstrated the ability of a very simple building block, this diphenylalanine, to form well-ordered structures related to amyloids that are associated with the degenerative disorders, and fully understand the phase diagram and changes of these structures. On the other end, the other extreme is trying to understand another typical self-assembly process, which is the formation of virus particles. In the past, there were attempts to target this self-assembly process, because if you think about it, you just need somehow to manipulate these structures to achieve antiviral therapy, which is much needed. And this is also things that we have in mind, but this is just an appetizer. I mainly want to speak about amyloids and amyloid self-assembly. So amyloid formation is another self-assembly organization related to pathological disease, Alzheimer's disease, Parkinson's disease, type 2 diabetes, about 30 different human disorders, but also functional structures in bacteria and other microorganisms. This formation of well-ordered structure, this is a typical self-assembly process, and we are very much interested to understand the processes at the nanoscale in order to give mechanistic insight into this phenomena, but also develop treatment to manipulate the self-assembly process. And again, I spoke about it 2 years ago in CLINAM about our phase 1 clinical trials, which were already completed. We are going to phase 2 clinical trials with EG30, I think the really first small molecule modulator of molecular self-assembly at the nanoscale. So being interested in the molecular basis for recognition and self-assembly process, especially in the very early steps, just to manipulate this aggregation process in very early stage. And again, this is old stuff, but in our reductionist approach, we were able to identify smaller and smaller fragments. We were the first to show that a pentapeptide can form typical amyloid fibers. Then we showed that a tetrapeptide can form typical amyloid fibers, and suggested a model that is based on the interactions of aromatic moieties to get such ordered structures from such simple building blocks. Later on, again, this is old work published in Science more than 10 years ago, we're able to narrow down self-assembling structures down to dipeptides, the shortest you could get with peptides. And many years, which took us to the field of technology, we demonstrated all these wonderful properties of these dipeptides motif that were based on reductionist analysis of proteins that are related to the formation of amyloid. This is beta amyloid, the polypeptide that forms ordered nanostructures. In the case of Alzheimer's disease, we narrowed down the recognition interface, showed all kinds of very unique properties, mechanical properties for these structures, piezoelectric properties, semiconductive properties. If we think about the future, it might be that the formation of this structure related to semiconductivity, luminescence of these structures. Only many years later, so we demonstrated already in 2009 the ability of the core recognition motif of the amyloids to luminescent in blue in the around 450 nanometers. About 4 or 5 years later, the leading in amyloid research, Chris Dobson and Kaminsky, demonstrated that the same occur for the large proteins. So this is a proof for our reductionist approach to the formation, understanding the formation of the structures, and also mechanical properties of amyloids are well studied. And of course, there are many technological applications for these structures. But again, I spoke about it last year, but it got quite a lot of attention, so I will just mention it in passing. We moved from peptides also to peptide nucleic acid. This is amide backbone. Actually, it's peptoid for the chemist. It's not peptides, but these are peptide-like molecules with side chains that are related to the bases. And we demonstrated, again, the ability to form ordered nanostructures with them. But the very interesting, and this is not a model, this is the crystal structure. We demonstrated that what we envisioned really happens. We can have stacking like peptides do, but also Watson-Crick structures like you see with DNA. again, it was published in Nature Nanotechnology in April, got a lot of attention, and will probably have also some diagnostic importance, because we see there also new physical phenomena, which is the red edge excitation shift, which the color of the structure could be changed according to the wavelength that it's been excited. So this was quite extensive work on trying to define the smallest peptide recognition element. But recently we went into even smaller structures that can self-assemble into ordered amyloid-like structures. This is actually started as a control. We demonstrated that a dipeptide can form atypical structures at the nanoscale, and we speculated that it's the amide bond, the planar amide bond, is responsible for the formation of the structures. And then we studied whether a single amino acid, in this case phenylalanine, because we like aromatic amino acid, can form ordered structures. And quite to our surprise, the amino acid did not form ordered structures at the concentration that peptide do. But if you go to concentration as high as hundreds of micromolar, you get the formation of well-ordered structures, amyloid-like structures. For those in the field, you could see the typical THT binding and Congo red birefringence from a single amino acid, no peptide bond, just a single amino acid. These are well-ordered structures. You can see the electron diffraction pattern. So it was the first demonstration ever of amyloid-like structures made of a single amino acid. And we suggested that this may actually be highly relevant to a pathological situation, which is phenylketonuria. Phenylketonuria is an inborn error of metabolism disease in which the individual lack the enzyme that convert phenylalanine into tyrosine, and if it's untreated, you get the concentration of phenylalanine as high as hundreds of micromolar. In those concentrations, that we see the formation of the amyloid-like structures. Indeed, we could demonstrate that these single amino acid fibers are toxic, and as a control, it's not only the concentration of the amino acid, because if you take alanine, which does not form structure, you don't see the toxicity. We could raise antibodies against these structures, and the antibodies cause the depletion of the toxicity. So it's like the Koch postulate to really demonstrate that this is structures formed by a single amino acid may have the etiology in this disease. Luckily or unluckily, we had tough referees, so we had to demonstrate it also in human tissues. And again, we went to a tissue bank in the Netherlands and could get a tissue from from a deceased patient of phenylketonuria, and demonstrated the formation of the structure. For first demonstration that such structure could be formed, and also positive staining with Congo red and with the antibodies. So this was quite paradigm shifting approach, but we wanted to see if this, and another thing just before getting to it, we solved the crystal structure also a short time ago, and what you could see, again, it's quite amazing for those of you who remember the structure of beta sheets. Beta sheets are organized like a zigzag. You have 1, 2, 3, 4, 5, 6. So here, it's non-covalently bound. It's the crystal structure of the single amino acid. And what I see here, and I'm trying to convince the scientific community now about it, it's something like a supramolecular, non-covalent bound beta sheet. So 1, 2, 3, 4, 5, 6, 7, 8. It's like a typical beta sheet made of amino acid without a bond. So if this was a surprise, we had another surprise trying to see if this is really true for other metabolites, and this is a paper to be published, just accepted last week to Science Advances, in which we tested other metabolites, and our approach was that self-assembly of other metabolites could lead to formation of others in other metabolic disorders. And we suggested a new paradigm for metabolic disorders that is related not just to having a lot of structures that have been accumulated, but the formation of amyloid-like structures. And there are many metabolic disorders in which there are accumulation of structures, and both amino acids as well as a nucleotide. To make a long story short, and again, this is going to be published, we studied a long list of metabolites, very simple molecules, and we could see also in this case the formation of well-ordered structures at the nanoscale, THT binding, Congo red binding. So it's really the title of the presentation is of the article is extension of the amyloid hypothesis. We suggested that such simple structures can form typical amyloids. Also with these cases, with adenine, tyrosine, orotic acid, phenylalanine, which we already demonstrated, uracil, we demonstrated the toxicity of these structures. Of course, alanine is not. It's not the concentration of the amino acid, but the formation of the structures. Moreover, we demonstrated that toxicity is not just a simple necrosis, but order apoptosis, as you see with amyloid structures. So here you see, and the late apoptosis you see with adenine, you see with cytosine, you see with tyrosine, you see with orotic acid, phenylalanine, uracil, and not with alanine. So we gave the demonstration that it's the formation of the structures and order assembly. So just to summarize in the 30 seconds that I still have, we demonstrated that ordered amyloid and other nanostructures could efficiently be formed by very short peptides, aromatic peptides that show as dipeptide, or have all kinds of remarkable physical properties, mechanical, optical, piezoelectric, semiconductive, so on and so forth. And this is true also for the physical properties are now being observed also in the amyloid structure related to disease. We can make amyloid-like structures by non-protein metabolites, and the metabolite assembly share also structural and cytotoxic properties with peptides amyloid. It's just a new way to treat metabolic disorders. Again, we discussed here patents, and we just got a patent, again, 2 or 3 days ago on the PKU, and hopefully get new therapy for this. Of course, this is the work of many people. You see here to represent the chemistry approach to clinical nanomedicine from the periodic table of my lab. I move very fast, and so if somebody is interested in more data, you can visit my website, as you can see here. Thank you very much.
Thank you indeed very, very much for this great talk. I was in my mind still with Dampere when I said that and wanted to make an allusion, so I'm very sorry to call you a bad boy.
Okay, you know.
But it was a great
we
talk.
are placing, so Danny is coming, all the things that I did in the past, Danny is coming a few years after me, so I feel very, and I highly like him and support him, so it's